as a volunteer. And I think, Kayla, were you going to talk about HMMR attack?
Yes.
Attack. And I don't know, did other people want to present anything? I'm not sure who PMTM Book 15 is. Jennifer or Marcel or Vince?
I was planning just to listen. This is Vince. I'll think about whether there's anything I can present to help with this.
Okay, that's no big deal one way or another. We can listen to Kayla and then maybe have a little bit of a roundtable discussion. Shall I switch over, Kayla, to you?
Sure. So let me just get myself in the proper workspace here and share my screen. All right, so I was just going to talk about another project that I had worked on. Martin, in one of the earlier presentations, talked about a project that I worked on doing bulk RNA-seq differential expression from FASTQ files using DE-seq2, which was my first exposure to AnVIL and developing workflow scripts and things like that. This was my second project involving AnVIL, which was brought to us with someone else in our department has this Java application HMMR attack, which I have just his GitHub here that has all of the code. So it's available on GitHub, but the idea was to be able to do this type of analysis. At one point, we were hoping for an R package, but then we kind of moved it over to AnVIL to be able to have the data and the scripts all in one place. So I can kind of walk through how I kind of worked through this and how it can be utilized and things like that, just as kind of a real world example. So Talayu did a really good job of kind of walking through if you were to do this kind of command line. You would need SAM tools, you would need their Java application, and you would run all these commands, and then at the end of the day, you're looking for certain output files from the HMMR attack application, and then you can run analyses on that. So the idea was to kind of put all of this into a workflow script in AnVIL. So they also wrote a paper, which was really helpful to grab real life data and move that into AnVIL. So going through just really quickly, if I can find, they used 6 of these SRA excision numbers, which I was able to download. And I actually, looking at the actual study, there were 12 samples, so I extended it out to 12 instead of 6. But the first step was to get that data into AnVIL. So I would just start up an RStudio session, and I would download the FASTQ files for each of the samples and copy them over to the Google bucket. So then that way, under our files data directory, we have all of the FASTQ pairs for each of the samples in the study. So data's there. That was a good first step. Copied it into our bucket so that it's available. And then from there, I started looking at what are the actual steps and what do we need for that. So the first thing is they assume that you have a BAM file. So I needed a BAM file. And that would go into probably our scripts. I can just kind of show how I created scripts from there. So first step was to get a BAM file from something like a BWA mem And then we need to obtain these .bam files. So that was my first step, which maybe it has been BAM index, and this was the command that I came up with. Martin was gracious enough to create a Docker image for this that would have SAM tools, Java, all of the applications that you might need. So that was the Docker image that I based it off of. And then I set various memory, CPU, things like that. So this task is performed once because we only need a single index file. This reference genome, again, I came to the paper and I realized from the paper, the reference that I grabbed, reference genome I grabbed and copied it over to the Google bucket was this GCF GRCH38, so a human genome reference genome. So I was able to download that file, copy it to the bucket so that we had that available to create this index. And that was only run once. And these were the different output files that I needed to go on to the next step. And then from there, this task, HMR attack run, is pretty much all of the steps that Hal laid out on his README, which I will kind of go through. We needed to create a sorted BAM. We needed to create a sorted BAM BAI file. We needed a genome info file, and then we actually run the HMR attack JavaScript and such to get these filtered gap peak files, filtered summit sped files, and things like that. So this command actually gets run on each of the paired samples in parallel. So this was run over the 12 samples, but creating like the BAM file, the sorted BAM BAI, genome info, running the Java application, and at the end of the day, what were we looking for was the filtered peaks, gapped peak file, the sorted BAM BAI, and the summits, because we were going to show various different types of analyses that you can use with Bioconductor packages. So I saved these 4 files for each of the paired samples. And then this workflow kind of defines our inputs, the calls to the various tasks. This scatter fastq pair is how you're able to run tasks over like the pairs instead of just over like one file or something like that. It was able to pair up the fastq1 and fastq2 files to be able to run that. And this took quite a bit of time to actually get working. We grabbed a smaller of the paired samples to get the script running and kind of debugged that way. So I would run the script on just a single pair, make any changes to the script, and then once I knew it was running on a single pair, I would then bump it up to all 12 samples. And then there was just some tweaking we had to do with memory and things like that. But after many attempts, we finally got it working, making a few minor tweaks and things like that. But I can kind of show you, that was not where I wanted to go. I wanted to go to Job Manager and just show kind of timing on how this worked. Timing graph. So this was the first, so if I kicked it off at like 11 o'clock, 11:35 apparently, this was the first task, the BAM index task, that only happens once, and that creates the index from the reference genome, and that took 1 hour and 45 minutes to run. And then once that completed, then it was able to kick off the second task, which was the HMMR attack run for each of the 12 samples, and they were able to run in parallel. And you could see that these ones were probably smaller size samples since they only took about, hour, 2 hours to run. And then some of our larger samples took anywhere from 9.5 hours to run. And then once that completed, we have all of in our workflow when we were defining our inputs and things like that. This would be the table of the FASTQ pairs. This would be the reference genome. But then in the outputs, we decided to set these as defaults. So that way, these locations would be saved in the participant sets table. So once this completed, if we went over to the participant set table, we could see that we have the location on the Google bucket saved for each of these files that we saved as outputs. They kind of save not in the best way when it comes to the like in parallel saving files and things like that. It comes out as like an array of locations, but these are the 12 locations for like the sorted BAM files and all of those for each of the samples. So then we could pull that information from the participant set table and then actually start doing some analyses. First thing that I did was grab the participant set table, filter for the last IG, and then was able to perform some QC using the attack-seq QC package in Bioconductor, just to make sure that the alignment was of good quality. And then we can kind of get a little bit deeper into some plots and things like that. I demonstrate using diffbind to identify sites that are differentially bound between two sample groups. So in the experiment, they were looking at some CD4 positive cells on day 1, 2, and 3. So we were going to do this diffbind on like day 1 versus day 3. The R-Great package reads in peak files and infers which pathways are enriched using functional enrichment analysis. So I demonstrated that a little bit. And then the ChIP-seeker package performs statistical testing of significant overlap among ChIP-seq datasets. So this is where the filtered peaked, gapped peak files come into play and doing various plots and things like that. So the idea is you get the data somewhere that you're able to, like onto the Google bucket, you're able to write the scripts that access that data and run whatever type of analysis you're trying to do. And this one was, something that ran for 9 hours, so I could just push the button, run the analysis, and come back hours later, and it ran and was done. And the output was actually saved onto the Google bucket. And then from there, you can take the output from from your script, and you can actually use it in the notebooks to demonstrate how you might use it in to do a type of analysis of some kind. So I guess that's everything I have to share for that. Just kind of a basic, didn't want to go too in depth of the background of HMMR attack and things like that, but how you might use it in another way.
Yeah, really,
That's
great.
really
Excellent.
nice. Seems like there's a whole cornucopia of methods upstream and downstream that are all now packed into this workspace. And it'd be, on the one hand, we could say, well, this is an ATAC-seq workspace, and surely people will who are interested in that technology will want to look at that. But I just wonder, is Anvil capable of directing people to all the stuff that you've done here? There's probably many different tags that you'd want to push up to the portal so that your workspace would be found when people are thinking about doing things like diffbind or other biologically interesting things that you've done there. Have you thought about maybe writing an F1000 research paper to expose this at a more, I mean, on the one hand, there are all the notebooks that you've got there, but it sounds like, even more narrative would be important and then to bring that narrative into Anvil so that people don't have to go finding it. These are all challenges that I don't think the Anvil project is ready to take on yet.
Yeah, I think, so the original motivation was obviously we were working with Tao, who's also part of the IoT and Moonshot project. So this was kind of working with them, but utilizing Anvil to be able to kind of bridge the two together. But I think exposure would be great. And I don't know the best way, because it's not like an R package that you can just say, use my R package or whatever. So yeah, I think if there were ways to get that out there, that it may be useful to other people. I'm not familiar myself on how to do that, but would be open to looking into things.
Did you get a cost for this whole project?
I did not. I don't know if Martin has a cost. I just kind of ran up the bill, I'm sure, but Martin would probably know more of it.
You spent less than $100 on the project. I'm not exactly sure what, but
Including all the false starts, so to speak.
that's right.
Yeah. So we could run Anvil billing over this thing probably if we got the right identifiers, and you could get the numbers. That seems like an important thing for a very intensive bit of work that you've done to know. It sounds like it was a reasonable cost for that. But of course, if you did it on your own machines and you were able to do it on your own machines, it would be essentially zero. So that's the kind of comparison I think we need for the population in my lab, where people are saying, well, we need to use the cloud. Well, why don't you try Anvil? Well, I don't know. I may wind up run up a big bill, and so I won't do it. And so getting data like that, which is to say, listen, this really intensive analysis was done. Here's what it cost, would help motivate people to come in. I'm blabbering because I have to leave at 12:30, so I'm sorry about that. But I wanted to make sure I, made a couple of points.
And I'm sure there's ways that you could, in theory, if you could do a lot of stuff locally too to help. I think some of those false positives, you can do a lot of the Cromwell workflow script development locally, and then you're not doing as many of the false positives. But I think we were running into a lot of memory issues setting the memory in the CPU, which is kind of why we had a lot of those false starts. But that's when you start, raising the bill a little bit, is when you have more computing power and things like that. So I definitely think there's ways that you could work around, but like you say, it's not terribly expensive for the amount of work that was done. Yeah.
I guess it's also fully reproducible now, so and other people can stand on your shoulders and either do exactly the same analysis on their data, or they're in a better position to tweak it, to customize it for whatever their analysis is.
Quick question before I bail out. Is there an ITCR connection of this work at all?
No.
There's also this NCI work group on containers that we get emails from occasionally. And I wonder whether, it might be an interesting forum to present this, because you did base it on a container, right? And then there's it's a cloud and containers working group, so
Yeah, interesting idea.
might be something for us to put our heads together and say Bioconductor and Anvil tackle a lot of the things that your group is working on.
One thing is it's really easy to share the workspace with a defined group. So Kayla could share it with everybody who participated in the workshop with basically one trip to the graphical user interface. But it's not so easy to share it with the world. You have to actually say, make my workspace available to the world, to somebody. I don't know who ever done that.
What about making it? I mean, I know we're talking about Anvil and we're working for Anvil, but it must be possible, let's say, to make this a sort of platform independent type of analysis. You have Cromwell, you have Whittle. Those things don't really require Anvil. If you wanted to be more inviting to a larger community, what would need to be done in terms of setting up a virtual machine, notebooks in some notebook service?
Yeah. In many ways, I think we should get more buy-in from Tao Liu, who wrote HMMR Attack, because we were kind of inspired by his work, but then it'd be good for him to come in and tell us that we've done everything wrong at this stage and that stage.
I doubt it.
And then also use his repository and his user base as a way of advertising.
Yeah. All right, I have to bail out. Sorry. Thank you, though.
Were there other things that people wanted to mention in the light of Kayla's presentation? I thought it was really great, Kayla, so thanks for that. Maybe I'll just switch over to our document here, and I realize we're a small group, but please feel free to talk. In many ways, you were the first group of people that I tried to use Anvil, present Anvil to from scratch, and I'd be really interested in hearing your first impressions of Anvil, and so that's like the first topic, and the second one is the workshops themselves, but maybe starting with just what people around the call thought about Anvil as an analysis platform or a teaching platform or whatever it was that you came here for.
Martin, I'll kick off if nobody else is chiming in. I'm not in the computing and IT programming realm, but learning about it. And so I very much appreciated that you all kind of threw us in and we were able to follow along and learn as we went. And there were very targeted, reproducible things we could do on our own. So thanks very much for taking somebody from nothing to something over a regular scheduled time over the weeks. I very much appreciated all your efforts.
Yeah, thanks for attending and being there, and I guess in some ways people did come back for most of the workshops most of the time, so it must have been satisfying at that level. Otherwise, they wouldn't have come back. So that's great. Have Marcel or Jennifer or the EMTM book 15? I'm sorry, I don't know. Do you guys have any? Have you explored Anvil at all on your own outside of the workshops, or maybe just have responses, from the workshop itself?
I think the workshops are really enlightening, and I'd be interested in using them for the classroom. So Levi's talk was pretty interesting. So I'd just have to get more information from Levi and how he set it up his institution, which is the same as mine. Yeah.
So would you actually use it in teaching, Marcel? Like to,
Yeah, I would use it for for intro courses. Yeah, because the setup is so much easier so that students don't have to email you with questions about how to install things on their machines. So yeah, it streamlines the whole teaching process.
yeah, yeah. It's kind of interesting because I don't really think that the Anvil grant really envisioned it. Well, it's kind of driven by these very large consortium scale data sets and this very large scale analysis. And at the same time, there's this obvious value in teaching, including reaching institutions where that might not have access to large scale compute resources. Um, so sort of a diversity and inclusiveness kind of element to it.
I've also enjoyed I don't know if you can hear me.
Yeah.
I've also enjoyed the workshops, although I haven't had time to go and explore Anvil outside of the workshops. I'm really grateful that these are recorded so I can go back and follow along. I think I really enjoyed like the last workshop and exploring these little details that I was thinking about how do things operate in Anvil and going through the package as I'm still really new to R. So it's really helpful that I get these little pieces of knowledge. So thank you for that. And also, I wanted to ask Kayla again, what was the language that you were using to write? Was it the common workflow language?
Yeah. WDL workflow. I'm not sure what it. Yeah, but WDL is the workflow script.
Okay.
That I used in both instances. I also think there's another one that you can use, but the only one that I've started using and have used is the WDL. But I think when you look into Dockstore, there's another type of workflow script that you could use if you wanted to.
Okay. Thank you.
Jennifer, do you envision how you might use Anvil in your day-to-day life, or is it still a little diffuse?
I think it's a little diffuse for me right now. So I'll have to think about that. But I very much want to participate in using it and supporting development where I can.
Yeah, it's great. Were there things that put you off, people in general, about using Anvil?
I can say at least for my exposure, at first it was very daunting to learn all of. All I can say is I feel like Anvil has so many moving pieces to it, but I liked how the workshops kind of broke down each of the aspects a little bit easier to understand and grasp. But I think that there is, when you're first starting out exploring Anvil and stuff, there is kind of a learning curve when it comes to, where is the data, writing these workflow scripts. Then you have to write Java notebook or Jupyter notebooks. You can open an R Studio, you can open a terminal, you can open R. Like there's so many things within Anvil that it just, it can be a little daunting and there is somewhat of a learning curve. But I feel like once you get the hang of where everything is and how it all comes together, I think it's pretty remarkable what you're able to do within Anvil. But I think a challenge for me was just, I see in the chat, like where to start and everything that there is in Anvil was quite a challenge too.
Yeah. 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 Vince mentioned also about how even after the first, week or so, um, we'd still be spending like a good chunk of each workshop just like getting going. And in many ways, the road had been made smooth for you. So if you're struggling, if you're tackling it yourself, it would be even more daunting. I was wondering, we should probably be, thinking about wrapping this up. There's a small enough group, but I was wondering if there were any specific suggestions, reflecting on the workshop. Maybe you would have liked to have seen more scientifically relevant material, or it could have been easier, or it was too easy, um, or in general, there are constructive comments about the workshop itself. Yeah, I felt a little bit like the science was a bit, it was sort of disconnected from the science. It was more emphasis on the technical aspects of doing things. It'd be really great to have this burning scientific question that happened to be easily solved by some little component of Anvil and then getting everything together. I'll say I actually like this pop-up format, the once a week for an hour over a kind of finite period of time. Um, I used to teach in a large undergraduate institution, and I really hated the semester style of course where you had to perform twice a week for a long period of time, and then you also felt responsible for people that they weren't learning or you weren't communicating, and in some ways I like these workshops where you can kind of present things and walk away and hope for the best and not be held responsible. So I like the shorter format. I'll also say that it seemed to like be a way of kind of generating interest. So I'd tweet things every week, and every week there'd be a few more people who expressed an interest. So I thought it was kind of effective that way. All right. Well, why don't we call it a day? And I'll just extend a thanks to you guys for uh, showing up today and over the course of the workshop. I think there should be a second iteration of these workshops. Give us a chance to refresh and everything. But thanks everyone for showing up. See you all in the Cyberverse somewhere.